Good afternoon, everyone. On behalf of whole team, I welcome all of you for today's webinar in the Data Science webinar series organized by Manav Human Atlas Initiative. Myself, Dr. Shailesh Kumar, moderator of today's talk. And I would like to inform you all that the allotted time for today's webinar is for 1 hour, out of which talk will be of nearly 40 to 45 minutes. Then we will have a question answer session of about 15 minutes. I request all participants to use QA tab, question answer tab at the bottom of your screen to put your queries. I repeat again, to use QA tab for putting your queries. You can put your queries in between the talk and which will be answered by the speaker during the question answer session. I request all participants to put their queries in the question answer tab only. All participants will also get a feedback form for today's session. And for those who are attending session through Facebook Live, will also get a feedback link at the comment box. So please be with us till the end of the session. Now I would like to introduce Dr. Madhusudhan, the speaker of today's webinar. Dr. Madhusudhan is working as Associate Professor at Indian Institute of Science, Education and Research, IISER, Pune. He did his PhD in molecular biophysics from Indian Institute of Science in 2000. Then he worked as a postdoc at Rockefeller University and University of California, San Francisco, with Professor Andrej Sali on molecular modeling. Those who are from bioinformatics may know Professor Andrej Sali. He also served as principal investigator at Bioinformatics Institute of Singapore and worked as an adjunct faculty at National University of Singapore and Nanyang Technological University from 2008 to 2013. And since 2013, he is working as associate professor in biology at IISER Pune. His research areas are computational structural biology, molecular modeling, computational biophysics, and bioinformatics. So these are the research areas. Now, without taking too much of your time, over to Dr. Madhusudhan for his talk on molecular modeling techniques and how they are used in designing, discovering therapeutics. Dr. Madhusudhan.
Thank you, Shailesh. And thank you, Manav, for giving me this opportunity to speak. I'm going to share my screen. And I'm guessing that it's visible to everyone now.
Yes, it is visible.
Yes.
Okay. So I'm going to talk to you today about molecular modeling techniques, and particularly how these modeling techniques could assist in designing or developing new therapeutic agents. Okay, and what I have planned for you today is to take you through the modeling techniques very, very quickly, because I actually teach a course on this. So I'm going to commit the sin of giving you a very broad and very general overview of these techniques. And then I'm going to use one case study that we did in our lab to try and design or discover therapeutic agents. And this is against the Nipah virus. Okay, and this was work done about a couple of years ago. And I thought that this is a very topical study given that we are engulfed by this pandemic. Okay, so computational methods of modeling that I'm going to talk to you about broadly fall into the category of what is called structural biology, more precisely computational structural biology. Okay, and variously, these techniques that are available now, the computational methods, can look at structures, the three-dimensional shapes of these different molecules, and they can look at these different molecules either when you have individual molecules such as these, or I don't know where to hide this. Put it there. Individual molecules such as single protein molecules, or you can have protein DNA complexes where you have more than one kind of biomolecule interacting with one another. And so the shapes become important because they give you an idea of how these different molecules actually come together and in some meaningful way perform some functional role in the cell. Okay, so it's important to know the structures. And just to sort of give you a better idea of this, let me show you why structures of proteins, for example, is important. So right through the talk, I'm going to be using mostly protein structures as examples. But whatever I'm saying here today applies in general to any biomolecule, because when you develop a therapeutic agent, you're essentially trying to get an end result where a molecule or molecules are targeted. That molecule can be anything. It could be DNA, it could be RNA, protein, anything of that sort. We know the most about proteins structurally. So most of what I'll tell you today is about proteins. Okay, but in principle, one can design algorithms and think of various creative ways of extending this to other kinds of biomolecules also. All right. Okay, so here is the example of why the structure of protein molecules is important. Okay, so let me show you this. Here is a structure of a protein in this multicolored thing over here, a surface representation, and you can see that it's a somewhat globular looking object. This is a protein. This particular protein is called MDM2. Okay, it's actually a very important anti-cancer drug target. And shown in this ball and stick representation is a molecule called nutlin, which is actually a lead compound or a precursor to a drug that targets this MDM2. And the idea is that if you inhibit MDM2 from doing its function, then presumably you can combat cancer in some effective way. Okay. Now notice that the MDM2 molecule has a kind of hole, and this nutlin, which is the lead compound, or the let's just call it the drug, is going and capping that hole. It's blocking it, and this hole is actually biochemically important for the molecule because that's how it performs its function. Okay. Now I'm showing you all this because the point I want to make is that the three-dimensional structure of the molecule is important, because as you can already guess, only when I know the three-dimensional structure will I know where the hole is, where the active site of the molecule is, or where the important residues are, or where the important amino acids are, and I want to plug that in some way, or I want to have my drug go and stick in that general region of the surface such that it inhibits the protein from doing its activity, or biomolecule from performing its functional role. Okay, notice that the region around the drug I have deliberately colored in purple, showing that these are the regions that are immediately neighboring the drug molecule. Okay, now let me remove the surface representation and just show you, and this is a representation that you may have seen in some textbooks and figures and all that, and these days you can even see it in the in the news when they discuss the proteins of the coronavirus. This is a what is called a ribbon representation. It just shows the polymeric chain of amino acids of a particular protein, MDM2 in this case, for folding onto itself, because that's how the proteins are structured. Now notice that the region that was shown in purple on the surface comes from various different parts of the protein. In fact, if I were to show you the sequence, so here is the sequence. I hope this is kind of visible. So, this is the sequence, and these regions, these purple regions can come from various different parts of the sequence. Now, the thing is that the different parts of the sequence come together in three-dimensional space, which you cannot otherwise figure out simply by looking at the sequence alone. Okay, and hence trying to get to the structure of these sequences is very important, because that is how you can figure out, where there is a hole and where you have to target your protein, etc. Okay, so this is the same thing with a different example, and the red molecule here is the equivalent of the nuclein that I showed earlier, and these are the neighboring residues, and you can see that they come from various different parts of the structure. Okay, now how do we predict the three-dimensional structures of these proteins, okay, or biomolecules? Now, I'll come to why we have to do the prediction in just a couple of minutes. But first, let me give you a very general outlay of a recipe to solve problems. And this is true not just in problems related to bioinformatics or biophysics, but this is a very general strategy. So you need to have a way by which you can represent the problem. Okay. Later you will see that my representation here is of individual amino acids. All right. And then you need to have a way by which you can score. You can assign some kind of a score to them, right? Just giving a very rough example, the score could be something along the lines of the interaction energy between different amino acids interacting with one another. Okay, and ultimately you need to use that score in some way such that you optimize the score. Let's say that you need to get as high a score as possible, or if you're looking at energies, as negative an energy as possible, such that the resulting structure is meaningful. Okay, because what we believe is that if you're looking at energies of a structure, then the minimum energy or the minima of the energy should be what the most optimal structure is. Okay, and this is what we believe from all the known structures that we have encountered in this universe. All right, so this is a general recipe, and anytime you encounter this problem, you need to think of how best you can represent the system, what kind of scoring scheme you will use, and how you will sample. Okay, and the overall objective is to reliably build the three-dimensional structure. Over here I've just said protein, but in general any biomolecule, starting from its sequence, or I want to reliably build the 3D structure of a protein interacting with a inhibitor, like a drug. Okay, so the emphasis is on the word reliably. So I need to have a way by which, after I build the model, I need to say, okay, here is a statistical measure of accuracy of this model, and if it passes a certain threshold, then I'm going to say I'll be satisfied with it. Okay, so when you build a model, you need to ensure that it is a reliable model. All right, so now why should I be doing these predictions computationally? Okay, why don't I take recourse to experiments? So this is the situation as of today when it comes to proteins. Okay, the situation is much worse when it comes to molecules like RNA. So we know of about 235 million protein sequences taken from various different organisms on this planet. Okay, and we know the structures from experiments for only about 30 if I'm charitable, 40,000 of them. So you can see that we are off by several orders of magnitude in our knowledge of the structures of these biomolecules. Okay, and even if I were to include computationally built models, then the number is, again around 10 million. So here again, we are off by an order of magnitude or so. Okay, so ideally, what we want to do is we want to grab hold of the experimentally determined structures, learn something from them, develop algorithms such that we can build these computational models, and along with experimental help, we'll be able to resolve the 3D shapes of all these molecules. And once that is done, presumably, we can use it for trying to design or develop drugs. Okay. All right. So there are two major classes of methods that are used in trying to predict the 3D shapes. Okay. The one that I will tell you about first is the method that makes use of evolutionary information. Okay. And the idea is simply this, that over the course of evolution, what Mother Nature has done is that even though the sequences of different organisms, the sequences of genes or sequences of proteins that are equivalent between organisms may be different, they have diverged presumably from a common ancestor, as this tree over here is suggesting. Even though their identities are different, what is similar is the three-dimensional shapes of these molecules. Okay, so if you happen to know the structure experimentally of any one of these molecules, then knowing the fact that all these other molecules are also related to this one, you will have a starting guess as to what this molecule looks like. Okay, so this kind of method is sometimes also called comparative modeling, because you're comparing to what you know and then you are constructing a structure. Okay, and the second class of methods is called ab initio, because over here you do not necessarily make use of these kind of evolutionary relationships. You simply make use of the laws of physics, or you do some kind of fancy machine learning with all the known structures, and you try and determine what the rules of how these molecules come together are, and then you devise an algorithm, and the computer program hopefully will make a correct prediction of what the three-dimensional structure is. Okay, and this over here right now, as I am saying it, holds for individual biomolecules, just one protein alone, for example. Okay, so the first method, the comparative modeling method, has these kind of steps. First, you locate a suitable, you have your target protein, let's say, you want to build a three-dimensional model of this. You locate a suitable template, which means a other protein which is related to this protein, whose 3D structure is already available. Then you align the sequences of these two, and from the alignment of the sequences, you try and deduce where the individual components of this target sequence are with respect to one another in 3D space. Okay, I'll tell you about this a little more in the next slide. Okay, and then the next step is to build the model, and then finally remember that our buzzword over here was that we need to reliably build a model. So after we build the model, we have to check and see if the model that we have built is protein-like. If we're building a protein model, or DNA-like, it should look like a bDNA, should have the appropriate number of Watson-Crick base pairings and so on. Okay, and in the case of proteins, it should have a hydrophobic core or something like that, something that makes it look like a protein that will convince you that you're probably on the right track. And if the answer is no at the evaluation step, you can iterate at any one of these steps and keep rebuilding the model till such time that you get a satisfactory evaluation. Okay, all right. So Now, the model building itself can broadly be seen as one of optimizing uh, uh, restraints. Okay, so you must have encountered problems like this in these very annoying analytical or IQ tests, right? They'll say, there is a problem where 5 people are seated in a row, and let's call them A, B, C, D, E. A is wearing a red shirt, and B doesn't like to sit next to someone wearing blue shoes, and D always prefers to be flanked by people wearing a cap, and so on and so forth. They give you a lot of information in a roundabout way, and then the question is, what is the order of people on this bench? Okay, so similarly, when you're trying to construct a 3D model of a polymeric chain, and the polymers, let's say, consist of various different kinds of beads. Over here, I've just shown 3 beads. Now, either from a computation or from some experimental data, I can deduce some kind of restraint. I can say, either an experiment or some kind of a computation tells me that whenever I have a blue object, it is separated from a red object by 5 units in distance, and it's separated by green from green objects by 10 units in distance on average. Okay, actually, what I really want to get is, I want to get the distribution of these kinds of distances. Okay, I want to get the distribution. So it's 5 units on average, but in some it can be 3, some it can be 7, and so on. On average, it is 5, meaning that the probability is the highest for me to get it at 5. Okay, but it doesn't mean that I preclude the other distances, and so on. So I have this jumble, and then red itself has its own preferences, green has its own preferences, etc. Then I try and construct a model which is consistent, or which gives me the highest joint probability of all these different individual probabilities of these restraints. Okay, that's the general idea. And the ab initio folding methods, they don't necessarily have a starting template from which they can make these kind of deductions as to how the different objects are oriented in 3D space. They make use of methods such as deep learning and so on and so forth, and they get results. In fact, just about 2 months ago, a company which is a spin-off from Google called DeepMind used neural networks, and they made significant progress in trying to accurately determine the 3D structures of these objects, of these polymeric proteins. Okay, all right. So I've told you that there is a necessity for making these kinds of predictions because experimentally we know very little. Computational methods such as the ones that DeepMind has, it's called AlphaFold 2, for those of you who are interested to look it up. These would then help us accurately describe the 3D structures of these different biomolecules. Okay, and once you have described them in the structure, it has various uses. One of the uses is what I'm going to talk to you about today, which is to try and develop drugs or therapeutics. Okay, and there's a whole laundry list of things that you can do once you're armed with the structure. You can determine what kind of function it does, what else it interacts with, and what role it plays in the cell, so on and so forth. Okay, all right. So the next step is to actually look at the drug molecule, because now what I described to you is the target of the drug molecule, right? So, for example, the targets of the drug molecules that are of very high importance today would be the proteins of the SARS-CoV-2 virus that's causing COVID-19, right? So, if I know the structure of this SARS-CoV-2, let us say spike protein, then I can target it with a drug or a therapeutic agent. Now, what kind of a molecule would form a drug? Okay, I already showed you one drug-like molecule, the nuclein molecule, right? And these are some of the desirable properties. It should obviously bind very tightly. It should bind, in fact, ideally so tightly that it will prevent, in the case of the spike protein, it will prevent the spike protein from trying to engage with, what is called the ACE receptor of the human host. Okay, and this will act as a stopper in between, and hence, presumably, the virus, after it enters the human system, will be unable to proliferate because the drug is acting as a barrier to its interaction with the human tissue. Okay, so a necessary condition, it should bind tightly. Okay, and the conditions for it to bind tightly is that obviously the physics of the interaction, it should complement the chemistry. Geometrically, it should fit in. You remember the hole that I showed you, it should plug the hole, which means that it's complementing the geometry and so on. And obviously, it should do it in a place where it will inhibit the protein from function. Okay, and we also like it to be specific, which means that, you can't, make it interact with a whole host of other target proteins, or much worse, some of the target proteins are from the host itself, which means that it will start inhibiting proteins which are vital for the human cells. Okay, and that is what causes drug toxicity. How do I take my drug molecules? Okay, and how do I find out which drug molecule is going to work? Now, there are various interact with the host protein is sometimes referred to as docking. Okay, and this docking is a computational method, and it can either be done by analogy, by comparison. I have already told you that, I can look at other structures that are similar. So suppose I had another similar protein which was bound to a drug-like molecule, then it's very likely that my target protein, which is similar to that template, would also interact with the same drug or some small derivative of that drug. Okay, so that is by analogy. Or what I can do is, I may not know what drug will act, but I have found the important hole in the protein, the active site or the binding site or the catalytically important region of the protein, and I want to target my drug molecule or my therapeutic agent to that particular region. Okay, and how do I know this? This is the important region because, either there is some existing expertise on this particular molecule, or I've done experiments and figured out that that is the region of the target that I want to use for this docking exercise. And of course, the last strategy, which is the most general of the lot, is blind docking, where I have absolutely no information as to what part of the protein is important. I also have no clue as to, which is the most appropriate drug. So I try a whole slew of them. I try various different regions of the protein. I see which ones I think are going to bind, and then I take it to the laboratory and have it tested. Okay. All right. So here is the same example that I showed you. Ideally, I want this to stick in such a way that it is tightly bound and it is specific, that this nuclein will target specifically the MDM2 protein and presumably nothing else. Or even if it is, it will bind so weakly that it doesn't have an effect. And it should not be toxic. Okay. All right. So the questions I want to ask over here is that where on the target will the drug molecule, which I've called the ligand over here, where will it bind? And which is the one that would bind? And remember that when I am trying to find out what drug molecule best binds a target, I'm going to use the same mindset that I that I had earlier, which is of using a suitable representation and an appropriate scoring scheme, and to optimize the scoring. Okay. Now, in this case, the scoring would be to try and figure out the energetics. Which is the drug molecule which, when bound to the target protein, gives me the best free energy of association, let us say, and I want to computationally estimate this. All right. Okay. Now, some of the desirable aspects that I need for my computer program is that it should be quick and efficient. Why quick and efficient? I'll tell you this when I show you the example. But the number of drug-like molecules runs into hundreds of millions. Okay, so whatever I do has to be fast so that I can rapidly go through an entire slew of molecules and then try and figure out which are the ones that are most likely to bind my target molecule the most selectively and most tightly. Okay, and I also need to have accurate scoring schemes, because the identification of the drug molecule is from a set of a few millions, like I said. Okay, tens of millions or hundreds of millions, right? So if my scoring scheme is not accurate, then I'm going to start getting a whole lot of molecules which are not really going to be drugs. And when I go to test them out in the lab, I will mostly be faced with disappointment. Okay. And right at the very end, I'm going to make a comment over here about the current state of the art when it comes to the accuracy of these scores. All right. So I've already said this, that I want to ultimately compute a brief free energy of binding. So the way the docking scoring schemes are constructed is that either I make use of physics, which I've called molecular mechanics over here, or I suggest some kind of an empirical scheme or a statistical scheme. Okay, and whenever I say statistical scheme, keep in mind that all the kinds of machine learning algorithms that one makes use of these days, all fall under these kind of statistical schemes. You have n number of samples, you learn something from there. How often does A interact with B, or A interact with C and not interact with D, etc., and you then want to apply that. So this is what I call statistical learning, knowledge-based schemes. Okay, so let me show you very quickly a movie of what the binding or what the docking program does. Okay, this is an example of a small molecule called biotin binding to a protein called streptavidin. Okay, now the computer program computes an energy score. Okay, notice that the hole or the binding site over here has been occupied by two ligands. One of them is the actual ligand, as has been shown in an experimentally determined structure. And we are trying to play the game where we are pretending that we don't know what the biotin bound streptavidin structure looks like, and we are using our docking program to suggest where it could possibly bind. And what the docking program does, and this is a example of blind docking, which means that when I start the movie, you see that it's placing it all over the wherever it can, and associated with each of these docking poses, as it is called, you get a different energy value. Okay, so if you will notice, the energy value when it is inside the site, and the green colored molecule over here is the one that was experimentally determined, and you will see that over the course of the movie it does not move, right? Whereas the other multicolored model, that is the one that is being docked. The computer program is docking it all over the place, and associated with each docking pose is an energy, and the one that gives you the minimum energy is the one that you will take. And if you go back to the start of the movie, you will see that the one that is closest to the actual binding site has the least energy. Okay, so in this particular case, the docking program was a success. All right. Okay, so let me tell you about how this is used, and this is the example that I'm going to talk to you about in the next 10 minutes, which is to do with the Nipah virus. So in 2018, there was a breakout of the Nipah virus in Kerala. So what we had done at that time is to take all the proteins of the Nipah virus, and there are 9 different proteins in the Nipah virus, and we said, let us try and see how much structural information is known about these proteins, and then if structures were not known, we will model them, and then we will use all these known structures and the model structures and do a drug screen and try and pick out compounds which we believe would be important inhibitors. Okay, right. So this was work done by a whole bunch of people in the lab. Their names are listed here. I'll show you a picture of all of them later. The Nipah virus proteome is shown over here. It has these 9 proteins. They're all assigned single letter names over here. N is nucleoprotein. N is nucleoprotein. M is the matrix protein. G is the glycoprotein. F is the fusion protein, and so on and so forth. And these were the known. If you look at this column over here, these were the only proteins for which I had known structures, and these other proteins had no known structures. So what we decided is that we are going to take on an approach where we will target every single protein of the virus, and ideally the strategy would be that you create a cocktail of drugs such that you have one molecule or several molecules attacking every single target protein of the Nipah virus. Okay, presumably that will help defeat the virus more effectively, and also may have some benefits to overcome drug resistance and so on. Okay. So what did we do? We did some comparative modeling, also called homology modeling. We built protein structures for those we did not know. Some proteins we knew only the partial structures of. We performed our modeling exercises. We extended these structures. Then we did our small molecule blind docking for these different proteins, the nucleoprotein, phosphoprotein, etc. Then we did some analogy-based docking for a few. And when we did the analogy-based docking, our small molecules were not drug-like molecules. They were small amino acid peptides. And we also dabbled a little bit in trying to design a vaccine. I won't have the time to talk to you about this, but in general, you should get the gist of how these vaccines are designed from what I have said today. Okay. All right. So, we made use of a program called Modeler to do the modeling of the individual proteins. We also supplemented it with an ab initio method. So Modeler is a comparative modeling method. We also use something called IISER to do some ab initio modeling. And if you compare it to the table previously, we now have models for all proteins except the protein C, for which we couldn't get anything, right? So from initially being able to cover 4 proteins, now we double that number, okay? And so, that is shown over here. So first order of business, for each of the proteins, using a tool that we developed, we could locate which is the most probable region on the protein surface where the drug would go and bind. Okay, and how did we locate this? The tool makes use of the fact that you have certain cavities on proteins, and we essentially tried to figure out from computations where these cavities are. And we also made use of a special feature of this cavity as to how much water it would allow into the cavity and so on. Okay, I can't go into the technical details, but suffice to say that we have one of the best methods in detecting these cavities. And these would be the places where we will do our directed docking, which means that we are not going to search all over the protein surface. We are going to take our drug molecule and slot it right in over here. Okay. And what is our drug molecule database? It consists of about 22,000, 23,000 different molecules, which is a non-redundant set of these molecules taken from a database called Zinc. Okay. And this Zinc contains, like I told you earlier, tens to hundreds of millions of molecules. And we selected a subset of already known drug-like molecules, because if you have drug-like molecules, then presumably you don't have to do that many more tests to find out its toxicity, etc., because somebody has already taken or presumably done most of these experiments with these drug-like molecules. Okay. Now we know the binding site because we used our algorithm to find it out. We have our Nipah protein, and now we make use of the docking algorithm. Now, to be on the safe side, we didn't use one docking algorithm. We made use of two docking algorithms. Okay, and these are two completely different algorithms developed by different people, different computer programs. And the logic was that though these docking programs are a little inaccurate, the chances that two of them will give you the exact same answer, meaning it will identify the same drug in the same binding pose, being incorrect, is somewhat small, right? So if two programs run independently, give you similar answers, then you have that much more confidence in the results. Okay, that's the general idea. With that, we docked various molecules, and here is an example of this, zinc. Zinc 94 something something. That's the name of the molecule according to the Zinc database, and we believe that this would bind the nucleoprotein very tightly. Okay, and you can see that the protein, the small molecule, the Zinc 9425 something, is bound by both the docking programs with the nucleoprotein almost identically. Okay, so that gives us great confidence that this is a very likely true positive. Okay, and what we then did is that we also designed some peptides that could go and bind these Nipah proteins. So here is a peptide that we designed that we believe would best bind this fusion protein. Okay. Now, this is a very important aspect for the Nipah virus, at least, because the fusion protein exists in two states. There's something called a pre-fusion and a post-fusion state. So our idea was that if we designed a inhibitor, which is a peptide, it will prevent this protein from switching over from one state to the other, thereby inhibiting the protein from proliferating in the host. Okay. Now, right in the beginning, I told you that the objective was to reliably build these models, right? So one measure of reliability is that even though we made use of two different docking software that gave the same answers, that gives me some kind of confidence, right? Another confidence booster for us is that, we performed molecular dynamic simulations, and we tried to estimate these free energies of the binding from these calculations. And you can see, even somewhat qualitatively, that over the course of time, and this is over some 100 nanoseconds of simulation, the peptide that we designed that would inhibit that fusion protein does not budge. It remains where it is. One sign of it being it being a weak binder, etc., is that it probably would have disengaged in the time that we did the simulation. Okay, so we are continually trying to assess how good our predictions are. Okay, and one other measure that we made use of, this molecular dynamic simulation, etc., calculating the free energy, that is based on the physics of the system. We have some enthalpy or an entropy to compute, and from that we deduce a free energy. And we calculate enthalpy by looking at hydrogen bonding and van der Waals interactions, making use of the physics. A second way of doing this is by looking at the statistics of these interactions. Okay, and over here we developed another method. It's unimportant, the name and stuff like that, but what it does is that it computes how often you would like to have particular kinds of amino acids. If you're looking at the peptide bound to the peptide inhibitor bound to the protein, how often pairs of amino acids like to face off against one another, and you can get some kind of a score from that. Okay, and the more likely it is that two amino acids like to face one another off, the higher is the score. Okay, so let me show you an example of tryptophan, for example. Tryptophan interacting with itself has a very high score, which means that they like to, if you have a tryptophan on one side of the, or a tryptophan-like molecule on your drug or an inhibitor, it would like to interact with the tryptophan on the protein, for example. Okay, and positively charged amino acids will like to interact with negatively charged amino acids, and so on. Okay, so from this you can get a statistical score, and presumably you can use the statistical score in some kind of an algorithm to give you an overall score for the interaction. The overall score is in some way standing in for the interaction energy. Okay, all right. So let me, before I summarize what I told you today, these are the various people who contributed to the work, and various people advised us on experimental measures, etc. So to summarize and to give you some take-home messages from this, the first thing is that any problem, especially the ones in bioinformatics, but this is very generally applicable, follows this representation scoring optimization troika. Okay, so given a particular problem, if you can fix the representation, intuitively select a good scoring function and then optimize it, then you probably are going to make good headway in solving a problem. Okay, we make use of this recipe in modeling the 3D structures of biomolecular complexes and biomolecules also, right? And these biomolecular complexes that I very quickly and briefly described to you today, were of protein-protein or protein small molecule peptide and protein small molecule ligand or drug-like molecules. Okay, I gave you an idea. I also told you that the assessment of these models is very crucial, because you need to trust what you have got computationally. You need to make it pass several tests of fire before you are satisfied with it, and then you need to validate it experimentally. So just for the record, what we did over here is that we got these different inhibitors, etc., for our molecule, from a list of, a few thousands of these molecules. We subjected it to various tests, statistical, empirical, etc. And we have now a set of about a dozen compounds on which we have the highest confidence. And were it not for the SARS-CoV-2 virus, we would have had these tested in the laboratory. But this is what we're going to do presumably in the next few months. Test our hypotheses, and then we will know for sure. And what I outlined for you today are exactly the first few steps that are taken by people in the pharmaceutical industry when they design or they try to discover drugs. Okay, and I keep saying design or discover because you don't always necessarily make these new molecules, but from an existing list of molecules, you try and see if something fits your particular target. Okay, and if you want to learn more about the particular example I gave you about the Nipah, that was published a little over a year ago in PLOS Neglected Tropical Diseases. Okay, so I will stop here. I know that I have committed the sin of compressing a lot of information within 45-50 minutes, but I will take your questions now. Thank you.
Sure. Thank you, Dr. Madhusudhan. It's really very interesting and very informative talk. And I know that this is very lengthy topic, and you just compiled it within 45 minutes. But I hope you could able to give message to the all participants what exactly the work is and how it is to be done. So there are few questions I have received during your talk. So I will take a few of them at present, and rest we will communicate to you via mail. So the first question, which is that what are the factors that should be considered while choosing a suitable template for modeling a protein through comparative modeling?
So usually you choose a template which is as closely related to your target protein as possible. Okay. Ideally, so let us say that you're looking at a human target protein, then and also you're looking at this as a target protein because no structure exists. Then you try and look for a closely related protein, something from chimpanzee or mouse or something like that, right? A mammalian source. But if you can't find it, then maybe you'll go to a yeast protein or something more distantly related. The more closely related the protein, the much better. The more accurate will be your model.
Okay. So is there any way to score them? Means
oh yes.
Means on what basis? Yeah.
Yes. So there are various ways of establishing similarities between sequences, right? Because over here you only have a human sequence. You may have a mouse structure, but ultimately you're looking at its sequence. You have values of significance of the relationship. For example, a very popular such measure is to use the p-value of blast, e-value of blast, right? So that gives you, so e-value is a number which the smaller it is or the closer it is to zero, the better is the closer is the relationship. And so that will actually give you a quantitative way of figuring out which is the closest template.
Sure. Thank you. There is one more question where they had asked, how do you recognize the site on the protein? Means if you say that this is the position where I have to dock this molecule. So okay, what are the ways while determining it?
Yeah, very good question. And I forgot to say something, which I'll say in answer to this. So currently, recognizing the site where you want to drug a particular target is a little bit of an open question. Okay. Now, I told you that we have developed a method that does this, but it's far from giving the correct answer every single time we apply the method. Okay. So ideally, what you would want to know is whether the site that you're targeting will inhibit the functioning of the protein, which means that you need to have intimate knowledge about the biochemistry of that particular molecule. And yes, this requires several times years and years of experimentation by different groups trying to figure out, where the binding sites are, active site, etc. Right? So, and especially when you do not know the structure, you see that these sites are actually built from the coming together in 3 dimensions of very separate parts of the molecule. So figuring that out from the sequence alone or from a model alone, because the model can contain errors, is a challenge. Okay. And the part that I left out saying, I alluded to it earlier, is that the state of the art when it comes to recognizing what molecule will actually bind this particular site is far from solved. Okay. In fact, if some of you who are listening, want to get a Nobel Prize for yourself, here is the problem. Figure out exactly how you will determine which small molecule will go and specifically inhibit a particular protein. Okay, specifically meaning it won't inhibit anything else. And if you can do this unfailingly, essentially your one computer program will solve, I don't know, like 85-90% of all human diseases, right?
Yes.
Just like that.
Yes.
Just like that.
So yeah, very insightful. So one more question, means that is obvious question. What are the softwares we used for docking, and is any free software available? And which is the best one? Best one for it?
Look, the one about which is the best is a little difficult to answer, because it depends on what data has been used to train or test that. Okay, but there is some global consensus about a few methods, which comes out in, there is an annual competition, okay, to try and dock small molecule ligands onto proteins and see which one does the best. Various people who have developed methods, and there are literally thousands of these methods, okay. The ones that we prefer to use are called dock and autodock. Sometimes we use something called ZDock, and sometimes we also make use of something called Haddock. Okay. These are all, we like most of these. There's something else called PatchDock also, which we make use of. These are all free. There are various commercial ones, which I think perform a little better, which is presumably why they're commercial. So if you have a little extra change to shell out, they would be, the software called Glide by Schrodinger. There's something else called Gold. So these are the various software. But there are many. And depending on who you ask, they'll list out their favorites. These are my favorite ones.
Yeah, sure. So yeah, some of them on which I also worked. So yeah, these are also favorite for many band competition.
Right.
Yeah. One more important question is that what about the energy of the molecule? Means if we say that potential energy or energy, whatever we are calculating, free energy, and we say that stable energy means minimum free energy is the most stable structure, but it is really active on molecule, or we are, if I talk about suboptimal fold or other thing, is there any effect of that energy level on the structure prediction? So how to solve this problem?
Yeah, so I won't answer the question kind of directly. Let me share. I'm still sharing screen, right? You can still see.
Yes,
right?
You can.
So let me tell you, when it comes to calculating the energies of these molecules, where the significant problems are. Okay, and I think that will put your question in or put the answer to your question in perspective. Notice that to ultimately to try and compute an energy, the energy that we really want is the free energy, either free energy of the structure or free energy of association if we're looking at binding. Free energies, we know this from thermodynamics, have a enthalpy and an entropy component. Yeah. Okay, the methods that we have, the physics and the statistical methods, etc., are very useful in computing enthalpy. Okay, we are really very poor at computing or estimating entropy. Okay, now there is a significant reward. I wouldn't say Nobel Prize, but it'll be a very significant advancement if somebody figures out a very smart way of accurately computing entropy of either a structure or an association, or whatever process you're looking at, such that you get a very good estimate of the free energy, right? So whenever we talk about energy, what we really want in the back of our heads is free energy, right? So if you want to write a computer program, you should be thinking, how can I compute free energy of whatever process, structure, or complex, or whatever. Does that put things in perspective?
Yes, yes. Yeah, sure. So one last question I'm just putting in front of you. That is that during MD simulation, the reliability depends upon the scoring mechanism which we use. Does using multiple scoring mechanism will help in credible result of?
Yes, so absolutely. Look, each of the scores, by now you will know, are imperfect. Each of the scores, are not exactly accurate, right? So if you get, multiple scores, and according to each of the scores, you're scoring significantly, or you're scoring above a certain threshold, etc., obviously it increases your confidence. Okay, so the bioinformatics people will sometimes like to refer this to this kind of a method as a jury system, where you don't rely on one score alone, but you assemble a set of jurors, 5 different scores. And then let's say that 4 out of 5 give you a favorable outcome. You say, okay, now I'm going to rely on it.
Yes.
Okay.
Sure. So yeah, thank you, Madhusudhan, for providing insights and answering the question. There are many more questions in front of me, but due to the lack of time, we could not take everyone. But we will send it through mail for your answers. So yeah.
Thank you. I do hope that, even though it sort of, I went at the speed of a bullet train, I hope that, people got a gist of, what needs to be done, and the complexity of the problem. I hope I was able to convey this.
Yeah, no, certainly we got something. Yeah. So yeah, thank you for this interesting and valuable talk. And I request all participants to fill their feedback form for today's talk. And those who have attended webinar through Facebook Live will also have to send their feedback through the link provided in the comment section. And thank you all for joining us for very interesting and interactive webinar. I really thank Dr. Madhusudhan for sparing time from his busy schedule for this talk, and with prime focus on the molecular modeling and docking part. So that is really very interesting. I would like to inform all participants that in the series of data science webinar, we have Dr. Pritesh from IIIT Delhi in upcoming days. So we'll have more webinars in upcoming days, and the next speaker will be Dr. Pritesh from IIIT Delhi. For more information, please visit Manav at social media platforms, Twitter, Facebook, and Instagram. So I would like to thank outreach team to organize this webinar and whole technical team for their support. Thank you all, Dr. Thank you all, and thank you very much, Dr. Madhusudhan, and good evening, all the best. Thank you.
Thank you.